Hello, everyone. It's an honor to be speaking with you all today. I wish I could be there in person, but I'm still happy to be able to share this information with you all. And thank you all for your attention over the next 30 minutes. My name is Trevor Murray. I am from Zaiput Flow Technologies. We're a United States-based company in Boston, Massachusetts, an MIT spinout company, and we work in the areas of flow chemistry and other flow technologies, some of which I'll be sharing with you here today, and so that you can learn about how these technologies can be applied to make your processing easier and your process development faster. So there's a tiny bit of background on Zaiput, the company. As I mentioned, we're an MIT spin-out company. We have commercialized a full line of extraction equipment. And we've grown over time to move just away from simple liquid-liquid separators, which is our true origin as a company, into more advanced technologies such as countercurrent liquid-liquid extraction platforms and fully integrated batch extraction platforms. I'm going to be focusing on these two areas today since they're kind of at the forefront of our development of the technology. Today, we really are an interdisciplinary team of engineers, primarily mechanical and chemical engineers, and we are looking to solve more problems in terms of processing chemistry and designing and developing new equipment for the industry. As the company has grown over the years, we've continued to both scale our technology in terms of both size of technology and in terms of complexity. Of course, the company was founded back in late 2012, so we've already been around for 10 years now. And over time, we've dramatically grown both in terms of our capabilities, the size of our team, and we are now truly an international organization with direct presence in the United States and Europe, and a fantastic distribution team spread across Asia, of course, particularly with Nippon Science Corps in Japan, who has made this talk possible today and will continue supporting the Japanese market for us for the long time in the future here. We sold technology in over 50 different countries, hundreds of papers, and we've sold on all 6 continents at this point, and we're still growing into new markets every day. But with all of that introduction out of the way, what we really focus on at Zaiput is liquid-liquid extractions. As you can see from the survey done back in 2011, liquid-liquid extractions are actually the second most common step in pharmaceutical processing, but it's a step that's often overlooked, right? A lot of time is spent focusing on our reactions, how we can optimize those reactions and reactor technology, but not much thought is put into this second most common step, which is extractions. And there are a variety of issues that have developed as a result of this in both batch and continuous processing. The issues in batch-based extraction are really threefold, right? The first one is a non-obvious problem, but a problem all the same, which is an issue of productivity. This first challenge really arises from the fact that when we are doing extractions in batch, oftentimes we will use our reactor at half capacity just so that we can leave space for our extraction solvent afterwards. This kills our productivity of that reactor by at least 50%, perhaps more depending on the volume of wash or solvent that you need following your reaction. Second issue is a time-based issue. For any n multiples of washes that you have to do, it's going to take n times the time. So if you have 2 washes, it'll be twice the time, 3 washes, 3 times the time, and so on and so forth. This once again really hurts our productivity in the plant because now we are using limited reactor capacity to carry out these time and labor-intensive extractive steps. Finally, another batch-based challenge have emulsions. Now, emulsions and other slow to settle systems can be because of small density differences in our system. It can be because of particular molecules having surfactant-like qualities. And in any of these varieties of systems, the slow to settle emulsion really, once again, slows down our ability to rapidly move molecule through our processing. We've worked with folks who have emulsions that take anywhere from hours to days to settle. We even had one customer who had an emulsion that took over 2 weeks to gravity separate out. And that's simply unacceptable, whether you are at the lab scale or at production scale. Now, of course, we are called Zaiput Flow Technologies. We come from the world of flow processing. And the dream in truly continuous processing is to put your reagents in one end of a tube and have your product come out the other end of a tube. Now, in reality, there is no solution to these intermediate workup steps, whether it be an extraction or a separation. So we ended up seeing these somewhat semi-continuous processing where the reaction was done in flow, but then the workup was done in batch. Now, that, of course, is where our technology comes into play. You can see it in action here. We have a biphasic stream entering the separator at the bottom, and then 2 completely separated streams coming out of the separator at the top. This is a simple water toluene system, but it does a good job of showing how simple and easy it is to use the separator. You really just start flowing into it, and it does the work for you. Now, of course, the question becomes, how does this all work? So the separator really has 2 components working together to actually effectuate the separation. On the left side of the separator, you can see a dotted line, and this represents the membrane inside the separator. Now, this membrane is not a traditional membrane in that it's a size exclusion-based membrane. Instead, it is a membrane in which we use wetting properties to control the separation. And by wetting properties, I mean hydrophobic, hydrophilic type interactions. On the top, you see a hydrophobic membrane, and on the bottom, you see a hydrophilic membrane. Now, what we see is when we drop water on the hydrophobic membrane, it beads up and is not allowed to pass through. But on the bottom, we see the water really soak into that membrane. And if we now had a pressure differential across that membrane, we would be able to push the wetting phase through the membrane, but not the non-wetting phase. So that is exactly what our differential pressure controller does. That is represented by this curved diaphragm here, and what this does is it controls the differential pressure across the membrane, providing enough pressure to push our wetting phase through without providing so much pressure that our non-wetting phase passes through the membrane as well. Now, this type of membrane separation has a lot of advantages, and one of the primary advantages is in the separation of emulsions. So you can see in this video here, we have a well-emulsified system in the beaker, where we have, this is an ethyl acetate, isopropanol, a vegetable oil, and water system. If you mix these all together, they emulsify really well because of the fats in the oil and the lowering of the interfacial tension by the isopropanol. As you see, it's a homogenized purple in the stirring beaker here. We take that, we pump it into our separator, and we have completely clean streams coming out. This is our organic in red and our aqueous in blue. And the reason that this separation is so effective is what we do is we take the continuous phase of the emulsion and we push that through the membrane. Now, what this does is this allows the dispersed droplets of the continuous phase to be carried through that separation chamber. They're still not going to be allowed to pass through the membrane. And so they are going to coalesce or come together at the same exact moment that we get our phase cut. So in this manner, we get very effective separation of emulsions, much better, of course, than batch-based systems, but even better than other sorts of separation technologies like centrifuges. Now, there's a host of other advantages of the technology. One of the very inherent ones is scalability. We start at the lab scale at 10 milliliters, up to 10 milliliters per minute, and we can go all the way to production scale. At the time of this conference, we'll be right around when we have the release of our new large-scale separator, which will be operating at 40 liters per minute, or about 2.5 cubic meters an hour. And what's amazing about this technology is you can optimize your separations in the lab at this small scale, and the technology scales within 1% of performance absolutely linearly, which makes the process development quick and easy. Now, there's a variety of other advantages to this technology. It can separate liquids with the same density. It can easily separate emulsions, as we've already seen. It's easy to use, easy to maintain, easy to clean. And of course, continuous operation is inherently built into these devices. Now, we see this technology used in three main processing modes. One is flow chemistry. The next is hybrid processes, or what we call hybrid processes, which is applying continuous technologies to batch problems, and then also in multi-stage extraction. Today, I'm going to be focused on the second two application areas in hybrid processing and multi-step extraction, since flow chemistry is something we've already covered a lot in our talks and our conversations in the past. And the reason that we are really applying this technology to batch processing and batch challenges is to solve some of these difficult separations and problems that we talked about earlier in this presentation. So the first one, of course, is using reactors at half capacity. So what our customers now do is, instead of doing a traditional batch extraction, they actually use their reactor to full capacity, or however much capacity they need. And then they will pump that directly into our extraction platform, where it meets some solvent that is pumped in from the solvent storage tank. Oftentimes this is solvent storage tanks or IBC tanks. We then do inline mixing. We've done some extensive work on sizing mass transfer to get you to your theoretical equilibrium, meaning this is as efficient of an extraction as possible. You are going to be able to get as much mass transfer as if you did high-speed mixing for an infinite period of time. And this is actually another advantage that our customers often find. Many times people will undermix a system just so that they don't over emulsify it and cause the separation to take too long. Because of our platform and because of our ability to size this mass transfer properly, we don't actually have that worry, and we can get advantages in our partitioning and our mass transfer just based on this one fact alone. Then, of course, we have our separator built into the platform. We get an instantaneous phase cut, and we can move on with our processing, sending our solvent, our product to the next step, and of course, our spent stream to waste. Now, this really has a lot of advantages over a typical batch extraction process, right? Because typical batch extraction processes are very labor intensive and time intensive. So there's the time that it takes to add the second phase. There's the time it takes for mixing, there's the time it takes to wait and separate out, and then of course there's the time it takes to drain and add the second phase after this. Now, in an ideal scenario for a 1,000 liter reactor, this is going to take about 2 hours. And for many of the customers that we work with, 2 hours is actually rather fast. For many people, this can take even a much longer period of time. If you take this and put it into the context of our extraction platform, it can be dramatically sped up, right? The time it takes for addition is instantaneous as those two phases are getting pumped together. The mixing speed is compatible with the settling, so this means we are unreliable on on our mixing speed. We just size out the mass transfer to give enough residence time in this tubing to come to equilibrium. There's no waiting for separation, and of course you do not have to sit and watch the phase cut and try to determine where one phase ends and the next phase begins, because our technology will do that for you. So what this can actually do then at 2,000 liters an hour or 2,400 liters an hour is actually double your capacity in less than half the time. So we are really seeing productivity gains of 4x over our traditional batch processing. Now, this is what the platform looks like in reality to give you an idea of scale in terms of width. That is only about 60 centimeters across, as maybe a little bit closer to 70 centimeters, but quite a small, compact platform. It's built on wheels so that it can be easily moved around the plant, brought from one reactor to the next, so that it's not just a bespoke unit. It is really a versatile unit for processing across your plant. Now, of course, we have pumping integrated to the platform. The mash transfer is inline. We have static mixing built in there, and that's going to be specified to your process. Of course, we can over-engineer the mash transfer so that you have a solution that's going to be applicable across all of your different systems. Some folks always worry about solids when we are dealing with membrane type technologies. So what we often do to mitigate this concern is just put a sort of inline filter before our separator. Then, of course, we run through our separator and the outlet product streams are at the top here. So then you can, of course, move those on to your next processing step. And as I mentioned, this whole platform is designed to be very easily cleanable. There's lots of replacement parts if you're really concerned about carryover between batches, and much of the platform can actually be hand cleaned and hand inspected itself. Now, of course, this is also applicable to multi-step extraction. This is when you could have maybe a acid wash and a base wash. This could also be applicable if you need to repeat a wash twice or perhaps even 3 times. The concept remains the same. Of course, here we have depicted just the same solvent going into 2 different washes, but it could be, of course, 2 different washes as well. And so here we're really solving that challenge of the amount of time it takes for 2 or 3 washes to be done, because the amount of time and flow that it takes for 2 washes is the same amount of time as it takes for 1 wash. So your benefit in terms of the time that it takes to carry this out is multiplied when you have multi-step extraction for purity's sake. Now, of course, we already touched on the challenge of emulsions and how we can solve that problem. Our technology truly is a density independent separation solution and readily separates emulsions. So there's no more waiting for those to settle out. Another question that we get all the time is what happens if I have an emulsified layer in the middle of my reactor, say a 3rd layer, and our separators can handle those too. Generally, those are made of some sort of solids, which we will, of course, filter out. And then it's beyond that, it is just an emulsion, which our separator can readily separate out. So all of these concerns of and loss of yield to those types of 3rd layers or the loss of yield to emulsions can actually be resolved through our separation technology. Now, moving on, we of course have these advanced multi-stage extraction platforms. So this is taking that multi-step approach, but going even a little bit further and bringing this really into a counter-current extraction setup. So for those unfamiliar with counter-current extraction, what it looks like is flowing one phase in one direction and the other phase in the other direction. In this example, we are flowing an organic from left to right and our aqueous from right to left. And the advantage here is that through countercurrent multi-stage extraction, we can obtain much higher yields or much higher purities using a lower amount of solvent. And so we can end up at higher concentrations of our desired compound when we are carrying out an extraction. So when do you really want to do multi-stage extraction? There are essentially 2 scenarios where you want to do this. You either have a low or medium partition coefficient for a single compound that you're trying to bring into another phase, or you have 2 compounds that are very similar and are very hard to separate from each other. So we have a low, what we call separation factor. In either of these cases, extractions need to be repeated so that we can enhance the purity or the recovery of our desired products. Now, taking for an example, right, if we had a partition coefficient of 1, which means if you carry out an extraction, 50% of the product will end up in your solvent phase and 50% will end up in your raffinate phase. Of course, if you put 50% over 50%, that gives you a partition coefficient of 1, which is what we see in this base case scenario here. Now, if we take this extraction and we bring it into a countercurrent extraction type setup, with just 4 stages, we can recover 80% of our desired product. With 9 stages, we can recover 90% of our desired product. And this is by using actually the same amount of solvent. So we are gaining efficiencies really in terms of how concentrated our product is, and we're reducing our solvent use and waste, which helps us in terms of cost. It can help us in terms of green goals, so using less solvent, and it can also reduce our burden on downstream process steps. Of course, if you have less solvent to handle, it can make sizing those downstream steps much easier. Now, there's challenges in carrying out countercurrent extraction because there's not a fantastic method of simulating this very often, particularly when you have new molecules. The thermodynamic models can be very ineffective, or they can be misleading, and the accuracy of those models can be quite low. And if you're really looking to carry out these types of if you really want to determine what this is going to look like for you on a production scale, you need experimental data. Unfortunately, to obtain that data in batch, you have to carry out this set of liquid-liquid extraction experiments. That's 30 plus extractions just to get a single data point for a countercurrent system. Now, this is not a very time or labor efficient process. It can take a single chemist weeks to months to get a small data set of what our extraction could look like at a production scale. Now, of course, that's where Zaiput's technology comes into the picture. We have developed the smallest laboratory scale platform for countercurrent liquid-liquid extraction available on the market. There's only 3 milliliters of volume per stage, 15 milliliters in the entire platform. So this allows the user, the chemist, or the process chemist to obtain a lot of data very rapidly. You can see that on the lab scale, we've developed a platform with 5 stages of separation. We have these tubing coils here, which is for our inline mass transfer. And then, of course, we've integrated pumping and control. So all the chemist has to do is plug their organic stream in one end, their aqueous in the other, and start pumping at whichever flow rates they like. And we can very readily carry out that extraction within the platform itself. Now, we took our platform and we modeled this against an Aspen simulation. And for those of you who are unfamiliar with Aspen, it is a chemical engineering simulation software. What we did is we set that Aspen simulation to 100% efficiency. We ran some common extraction systems recommended by the European Federation of Chemical Engineers as easy to model systems. And we then ran this on our extraction platform and compared the results. And what we found was that the experimental results that we got on our platform were within the error bars of what we would expect based on the Aspen simulation set at 100% efficiency. So what does this mean? This means that our platform is getting you as efficient of an extraction as possible. It means that we're getting you to a theoretical efficiency at each stage. And the reason that matters is that means that if this process is optimized correctly, that makes scalability quite easy, because you may find that you need 5 theoretical stages at the lab scale. A theoretical stage is always a theoretical stage no matter which scale you are at. So when we go to a production scale, it is also going to be 5 theoretical stages of our need. Now, of course, our technology readily scales. We can design platforms with any number of stages that you like. You can see a pilot scale platform on the right here. This actually comes with industrial controls. And we can even go to larger production scales with this technology as well. Now, as a quick example of how useful this technology can be, we have a case study. This was done by or published by Novartis. This is a process in which they wanted to recover a variety of alcohols into a water stream and then recover a couple organics into a decane stream. And this was a process that was being carried out in a production scale, but they wanted to optimize it and determine how to really better carry out this extraction to really recover more of these organics from the aqueous stream. So they went to our technology and ran a variety of experiments. The results were exactly what you'd expect. As the number of stages were increased, the fractions of alcohols in the organic stream were decreasing, and similarly, the fractions of these various organics in the aqueous stream were decreasing. But what was really key for this process, or for this scientist, was that they were at steady state within 10 minutes. And so that allowed them to very rapidly obtain a lot of data on this process, and valuable data that could then be taken back and applied to their production environments. Another example of this, and this is actually scaled up. So we've started this work at the lab scale and scaled it up to the pilot scale, is the recovery of a phase transfer catalyst. As it comes out of a Corning flow reactor, we have a single separation followed by a 3-step countercurrent extraction to recover that phase transfer catalyst. Now, the real challenge with this process was that there was emulsion that took 19 hours to gravity separate out. And of course, if you were going to do 3 extractions of 19 hours each, this is a process that would take days long if we were going to be carrying this out in batch. But of course, with our multi-stage technology, we can readily separate these emulsions, and the emulsification was not a concern. Now, we're not the first people to invent countercurrent extraction. There are, of course, other technologies that exist, for instance, mixer settlers, centrifuges, and columns, each of which has its advantages and disadvantages. Mixer settlers are common just because of their low cost. This is where they have a significant advantage over other technologies. People go to centrifuges often when they have systems with similar densities or systems that they're having difficulty to separate, although sometimes centrifuges can actually make emulsions worse. Next, of course, there are columns. Columns really have their advantages in terms of suitability for multiple stages. But beyond that, they're a rather inflexible technology in terms of both flow rate, numbers of stages. It's not a very modular technology. But Zaiput's scalable extraction technology has a variety of advantages. Of course, solids can be a challenge with our membrane technology. We can mitigate this challenge by just pre-filtering a stream to take that challenge away. In terms of multiple stages, we've demonstrated this technology up to 10 stages, but there's no reason that we can't go higher. So it should work, but we, of course, in the name of honesty, can't say for 100% that maybe going to 100 stages would be challenging. But our customers really come to us because of a couple key aspects of an extraction. The first is easy scale up. Since we work at a theoretical efficiency, we know that what works in the lab will work at pilot and it will work at production. So our customers can very rapidly move through these scales. Of course, we've chatted a lot about how easy it is for our system to separate emulsions and liquids with same densities, but the true advantage comes in terms of internal volume per stage. Our technology is orders of magnitude, 1 to 2 orders of magnitude smaller than any other technology. So this saves you material, this saves you time, and especially for new processes or material limited processes, this is a significant advantage to the process chemist looking to optimize an extraction. And with that, I would like to come to a close. Thank you all for your attention today. If you have any questions, you can, of course, email me directly at tmurray@zaiput.com, or you can contact our team in Japan, that is Nippon Science Corps, and they're, of course, exhibiting here at Interphex, and will be more than happy to answer your questions at their booth. I hope all of you have a great rest of your day, and thank you again for all of your attention.